As we enter the holiday season, many of us look forward to celebrating long-standing traditions with family and friends, which is no different than from us here on the podcast. While not necessarily as long-standing as some of the other classic holiday traditions, after 2.5 years on air, we've established our own end-of-the-year tradition, which is to take a look back at some of our favorite papers or some of the more intriguing manuscripts published in the journal over the past year. And so you'll see today, thanks to the different areas of expertise and general interest amongst our panel, we've selected quite a variety of papers to discuss, ranging from evaluation of new blood culture systems to use of metagenomics for infective endocarditis, and to the potential application of interferon gamma release assays for detection of histoplasma infections. And so suffice it to say that there will be something of interest for everyone listening in. But for those watching today, you are clearly getting an extra special treat as you get to see us do this episode wearing our best holiday gear and accessories. Welcome to Editors in Conversation. I'm your co-host and JCM editor, Ellie Thiel, and as always, we are joined by JCM editor-in-chief, Dr. Alex McAdam from Boston Children's Hospital. Alex, how are you?
I'm fine. Are you going to ask me about my sweater? I asked you
Yes.
about yours before we started.
Yes, it is a lovely shade of red. Does it have a story?
Well, it's kind of pitiful compared to how bright the sweaters are that you and Trish are wearing. But this was given to me by my mother-in-law, who I'm very fond of, several years ago. She got one for me, one for my father-in-law, one for my brother-in-law, and it is a tradition to wear them on Christmas Day. And since you notified me that you and Trish had this great idea around 8 o'clock last night, I didn't really have resources to get hold of anything else.
So I have two thoughts. A way to get bonus points with the mother-in-law by wearing it on the podcast.
Good.
And also this was all Trisha's idea.
I was so excited when I thought of this. I'm like, do you think Alex will be in?
Oh, it's a great idea. I love it.
Totally in. So, for our listeners, we hope you've enjoyed previous editions of Editors in Conversation, and we ask that you subscribe and review this episode wherever you get your podcast. This episode is brought to you by the Journal of Clinical Microbiology, available at jcm.asm.org and on Twitter at jclinmicro. Editors in Conversation is supported by the American Society for Microbiology, which publishes JCM. And remember that if you have a paper accepted in JCM and are an ASM member, you can get up to 50% off of publication fees. All right, so continuing with the theme of tradition on Editors in Conversation, she's already made an appearance today, but Dr. Trish Simner from Johns Hopkins is once again joining us to discuss her favorite JCM papers. And Dr. Simner has been on this podcast a few times before, including on the last two and really only two other previous editions of the Best Of podcast. So welcome back, Trish. Thanks for joining us.
Hello. Thanks for having me back, Ellie and Alex. It's really an honor to be a part of this tradition with both of you and our new tradition with our ugly Christmas sweaters and accessories. Apparently, I need to step up my game next year, Ellie. So I'm excited to be here.
Yeah, this was rummaged from my daughter's room. But I would just like to say we are not always going to dress up for every Editors in Conversation podcast. This is strictly holiday time.
Let's see where it goes.
Okay, moving on.
That's kind of disappointing.
I am not ready for that level of stress. So, getting into the meat of the podcast today, so each of us has selected 2 or 3 JCM papers that we want to highlight from this past year, which I have to say on behalf of the 3 of us that it's always a difficult task given the quality of publications in JCM. So before diving into each paper, we'll also share why we chose that particular study. And so with that, I will turn it over to our guest. First, Trish, what is your first paper and why did you choose it?
Okay, well, I picked my papers with my 2 favorite topics when it comes to Clin Micro. So, of course, NGS for infectious disease diagnostics is one of my favorite topics. So I chose the article entitled Pathogen Detection in Infective Endocarditis Using Targeted Metagenomics on Whole Blood and Plasma, a Prospective Pilot Study. It was by Dr. Laura Florin from Dr. Robin Patel's group at the Mayo Clinic in Rochester, Minnesota. I think you know where that is, right, Ellie?
Vaguely.
So I chose this one. Again, there was a lot of high quality kind of NGS related studies in JCM this year, but because this is one of the first prospective cohort studies to test targeted metagenomics for the diagnosis of infective endocarditis, I also really liked how they evaluated the specimen type in parallel. So they looked at both whole blood and plasma. So we know that infective endocarditis has high fatality rates of up to 25%. And despite our best efforts, whether that be applying clinical, echocardiographic, histologic, microbiologic workup, whether that's pre or post valve surgery, many cases of infective endocarditis go undiagnosed, up to 40%. And so next generation sequencing methods have been evaluated for this purpose, whether they be targeted or untargeted approaches, and they have demonstrated potential, but they've really been incompletely evaluated at this point. So the authors of this article wanted to explore targeted 16S rRNA next generation sequencing applied to blood-based specimens to see their potential in culture-independent, non-invasive diagnosis of infective endocarditis. So as I mentioned, this is a pilot prospective study looking at the yield of targeted metagenomics assay performed on both plasma and whole blood for infective endocarditis diagnosis, and to evaluate factors influencing the test performance.
So Trish, maybe before you go forward, so this is focusing on targeted metagenomics. Do you want to kind of quickly explain the difference between targeted and untargeted and what the advantage is of targeted metagenomics?
Yeah, that's a great question and great way to kick us off here, Ellie. So targeted, especially in this context for infective endocarditis, allows, so as you know, overcomes the barriers of having to sequence a host nucleic acid, which in an untargeted approach, you sequence all nucleic acid from that specimen. So the host DNA can interfere with your analytical sensitivity. It also makes interpretation and the bioinformatics programs a little bit more complex. So using a targeted approach, you can potentially get increased analytical sensitivity and simplify your bioinformatics and interpretations of the results. From a disadvantage, the targeted metagenomics is only going to detect what it's targeting, right? Mm-hmm. Whereas with with untargeted approach like shotgun sequencing, you can detect additional factors like antimicrobial resistance genes or virulence factors, and you could detect non-bacterial pathogens, where with 16S, you're obviously only going to be detecting bacterial pathogens. So, which is an example, there is a great example in this study where there's a Candida albicans positive sample that obviously doesn't go detected by this targeted method. So
perfect. Thank you.
Yeah. So in the study, they enrolled 40 subjects. They were prospectively enrolled at an early phase of diagnosis with either possible or definite infective endocarditis. And they were consented to obtain 20 ml of whole blood specimen, which were then divided into 2 EDTA tubes, of which one was harvested for plasma. At the end, they had 35 patients total, of which 28 were definite and 7 were possible infective endocarditis cases, according to the modified Duke criteria, and included 6 blood culture negative endocarditis cases. So in terms of methods, they amplified the V1, V3 region, so the standard region that we use in ClinMicroLab for 16S rRNA gene sequencing, that generated a 530 base pair amplicon. It was then sequenced on the MySeq instrument, and the pathogenomics RipSeq software was used for analysis. The results were then interpreted by a clinical microbiology lab director with expertise in metagenomic testing. I think that's important. These results are never easy to interpret, whether it's untargeted or targeted. And those interpreting results were blinded to other results available for those samples. They were considered positive if the pathogen detected by the targeted approach was also detected by a blood culture or other microbiologic testing, or were a known potential causative pathogen of infective endocarditis. They did something interesting in the study. So if they were originally negative on first try, they then went through the whole process a second time using a different extraction method. So on first attempt, they used EasyMag. On second attempt, they used MagnaPure. So overall, the turnaround time for the process was either 60 hours if they got a result on first attempt, a positive result, and 120 hours if repeat sequencing was required. So what they found, do you guys have any guess as to whether whole blood or plasma would have been better? Any thoughts there?
I should have read the paper more closely.
It's okay.
All right. Why don't we just divide it up? Ellie, you pick one, I'll take the other.
I'm going whole blood.
All right, so plasma's mine.
Okay, so this is great because you both win because there was no significant differences between the specimen types. However, Ellie, did you say whole blood?
I sure did.
There was more positives detected by whole blood, so 20 positives versus 16 by plasma. However, there was no statistical significance. But when combined, they were able to achieve 23 diagnoses, positive results by the targeted metagenomic pipeline. The positivity rate was impacted by time since last positive blood culture. So all specimens that were collected within 24 hours of a positive blood culture were positive by the targeted metagenomics. And then the targeted metagenomics trended towards negativity the further out they were from the draw of the specimen from first antibiotic use. But the exciting thing that I think was most exciting about the results here is that a potential pathogen was identified in 5 of the 6 culture-negative infective endocarditis cases, which included a Kingella species most closely related to Kingella denitrificans, Bartonella henselae, so cat scratch disease, Cutibacterium acnes, a Corynebacterium species, and a viridans group strep. But they have found that sequencing the negatives twice, as well as including both plasma and whole blood, increase their positivity rate overall. And you might want to be asking, well, why plasma over whole blood, etc. The thought is that potentially whole blood, you'll detect the intracellular bacteria, not just the cell-free DNA by plasma. And if I would have had to vote, I would have thought whole blood would have done better than plasma. But it seems like so far in this smaller study, there was no statistical differences, although trending towards whole blood picking up a little bit more. But neither of them picked up all the positives. So overall, although this is just further study is needed, the results of this pilot study suggest that blood-based targeted metagenomics may provide pathogen identification in subjects with infective endocarditis, including in culture-negative cases. And when we consider most cases of blood culture negative endocarditis, it's usually the fact that it's negative is because of prior administration of antibiotics. And so it's a common known ability of these next generation sequencing methods to still detect the nucleic acid in the absence of a viable organism, and has been highlighted as a potential role of previously treated infections that you can use these. And this study further highlights that in this approach.
Nice. So you said there was one Bartonella. I'm curious, was there any Q fever cases in their patient subset, right? Because Q fever and Bartonella, we still often rely on serology.
Correct. Prior to valve surgery, you really just rely on blood culture, Coxiella, and Bartonella serology, right? In this case studies, there was no Coxiella positive, but kind of exciting that they did detect a Bartonella in it. So,
I think serology still has a place to stay, but that was really cool that they detected that. Absolutely.
That is a really interesting study. And Ellie, I congratulate you on turning the conversation to serology.
Hey, any chance I get.
Trish, did I understand correctly that basically they were doing, in many cases, testing 4 times, blood and plasma, and then if those were negative, doing them both again with a separate extraction technique? Is that right?
Yeah, if they were
Yeah.
negative. And actually, I didn't mention this, but another highlight of this study is that, one thing that I think we need to get better at is standardizing is our controls with these assays, but they
Mm.
did a really good job of having a positive extraction and sequencing control with Corynebacterium glutamicum, and then a negative control as their extraction buffer. But they also did an inhibition control, which they spiked in like had a separate sample in parallel that was spiked in with the Corynebacterium as well. So they
And
did excessive, a lot of
it's a lot.
controls and a lot of
Yeah.
additional testing.
Yeah. And am I right that with those 4 different methods, in the end, none of them stood out as the leader method? They found 5 or so cases where they got a diagnosis or found an organism where culture had not. But one of those methods didn't pick up all 5 or 6.
Correct.
They were okay.
Yeah, and you think about it, one of the most important things that of these assays is the extraction method. And, I think that's one thing that we need to evaluate further, as we're trying to get kind of these boxed kits for these NGS assays. The most important component is the upfront extraction. So we need to find better ways of controlling for that as well through the process. And most labs have different extractors, right?
Mm-hmm.
And so it's highlighted even further in this study that depending on the extractor, they might actually be able to yield an additional result in some cases.
When do you see this kind of testing coming into the mainstream in large academic labs like yours?
I mean, we're working on some of this currently, not from whole blood and plasma, but we're working on other sterile fluids because we've done metagenomic next-gen sequencing from CSF. So I think we're starting to see the uptake of these methods into larger academic centers. But I see the same transition as what we've seen with PCR-based modalities. It started out really in only large academic centers with the expertise. And then as we started to learn how to use it clinically, and as the methods became more and more automated, it's now kind of mainstream, right? I see the same trend that we're seeing with NGS in clinical labs now.
So Trish, I'm sure you could talk about this for hours and hours.
Absolutely.
But we're going to move on to some other topics. So Alex, I'm going to throw it over to you. What's the first paper you're going to share with us?
The first paper I chose is titled The Impact of Implementing the Virtuo Blood Culture System on the Characteristics and Management of Patients with Staphylococcus aureus Bacteremia. And this came out of WashU School of Medicine. The first author is Miguel Chavel, and the corresponding author and last author is David Warren. And I chose this because, like every clinical microbiologist and every specialist in infectious diseases, I'm interested in blood culture. It's highly significant. They're performed in very large volumes in all laboratories. And as a result, you can do a high quality study because you have a large N just by virtue of being in a large hospital. Another thing I really liked about this study is that they looked at whether changes in lab practice had clinical consequences for the patients. And obviously, clinical outcomes are the most important potential effect of changing laboratory practice. So in this retrospective study, they compared 2 time periods, each about a year long, during which different blood culture systems were used. In 2018, the pre-implementation period, the Versatrex system was used at Barnes-Jewish Hospital. And in 2019, post-implementation, the Bactigular Virtuo system was used. And I'm going to refer to these as pre-implementation and post-implementation from here on. The bottles used during pre-implementation, the blood culture bottles in pre-implementation, did not contain antibiotic neutralizing resin, and they depended on dilution of the blood in the broth to overcome inhibitory effects of antibiotics. In contrast, the bottles used post-implementation had a polymeric resin to neutralize antibiotics. They evaluated detection of Staph aureus bacteremia in patients 18 and over who had an ID consult, and they looked at antibiotic use and clinical outcomes as well. Patients were excluded if they died within 48 hours of the initial positive blood culture, or if they had recurrence within 90 days of that initial positive culture, separate from that first infection. The institutional practice was standard. They collect 2 sets of aerobic and anaerobic bottles, each with 10 mils per bottle from 2 separate phlebotomy sites. Speaking from a pediatric institution, that makes me insanely jealous. We get like 1 mil in an aerobic bottle. Repeat blood cultures were collected at a physician's discretion. They didn't have a protocol for that. After those exclusions, they had 416 episodes of Staph aureus bacteremia in 415 patients, 176 pre-implantation, 240 post-implementation. The clinical characteristics were similar in the two time periods, except there were more episodes of catheter-related bloodstream infections during the pre-implementation period. So the first thing that they noticed is that the median time to positivity for the cultures was shorter in the post-implementation period, 13.3 hours compared to 17.8 hours pre-implementation. And that's consistent with some earlier studies that were done in the same institution using the same system.
And Alex, that's just focusing on Staph aureus specifically, correct?
Yes,
Okay.
yes, everything is Staph aureus, right.
Okay.
All right, so this is where it starts to get interesting. The duration of bacteremia was longer post-implementation than pre-implementation. So post-implementation, the median duration was 3 days, whereas it was 1 day pre-implementation. Along with that, a larger number of patients post-implementation than pre-implementation had bacteremia lasting 3 to 6 days or greater than 7 days, so longer periods of time. For 3 to 6 days, it was 40% versus 23%, and for greater than or equal to 7 days, it was 14 versus 44% for post and pre-implementation. So the bottom line, Staph aureus bacteremia appeared to last longer with the new blood culture system. And the authors suggest that differences in the blood culture media could explain this, with neutralization of antibiotics in the post-implementation period allowing bacterial growth despite the presence of those antibiotics. And that interpretation certainly seems sensible to me, and I can see Trish nodding.
I'm also nodding.
Excellent. But you have the green sweater and Trish has the red, so it's easier to see her. So what were the clinical consequences of that prolonged detection of Staph aureus bacteremia in the post-implementation period? You might expect increased testing for bacterial endocarditis in the post-implementation period because the proportion of patients with prolonged bacteremia was higher. But it turned out that the use of cardiac echo exams in most imaging studies was unchanged between the two periods of time. There were slightly more studies that were done to look for metabolically active malignancies in the post-implementation period, but it's a small increase, and frankly, it's hard to know what to make of it. I didn't think it was really likely to be linked to the blood culture systems. There were some changes in the use of antibiotics. These changes were in the definitive therapy, meaning the therapy that was used later in treatment of the infection. Vancomycin was used for definitive therapy in 35% of patients pre-implementation, but only 22% post-implementation, and that difference was significant. So there's less vancomycin. What was there more of? What there was more of was combinations of two antibiotics. Those were used in 6% of patients pre-implementation and 16% post-implementation, which was another significant difference. And just for those who are curious, the most common combination was daptomycin and ceftaroline, but there were 4 or 5 different combinations that were used. So one possibility is that combination therapy was used in response to what was seen to be prolonged Staph aureus bacteremia, which was more common in the post-implementation period. They didn't have a way of directly assessing that, but it certainly makes sense. The proportion of patients undergoing procedures for source control didn't change. Length of hospital did not change. Readmissions, mortality did not change between the two periods either. They make some interesting points in the discussion. Previous studies using blood culture systems other than Virtuo have linked time to positivity in blood culture to mortality at 90 days. They did not see that in this study of Staph aureus. They suggest that time to positivity and the duration of Staph aureus bacteremia reflect the blood culture system used, as they saw, and so that should be considered when interpreting this kind of study. Along these same lines, they point out that there are some studies on the importance of the duration of bacteremia that don't even specify what blood culture system was used, and they probably should have. So I thought this was a very interesting study. We think a lot about how changes in the lab may affect patients downstream, and this is a very concrete example of how it did change use of antibiotics, but the other clinical parameters they looked at were unchanged.
It's so interesting. So did they not evaluate kind of whether additional antibiotics were added when there was repeat positive blood cultures?
I wondered the same thing. Yeah, I'm sorry, Trish, go ahead.
Yeah, because, in thinking through it, you think that the upfront definitive, the initial therapy, once they knew it was Staph aureus and potentially, and the AST profile, that probably wouldn't have changed too much. It's more that there was this, repeat positive blood cultures making them think that there's a chronic source or, prolonged bacteremia, what's going on. And so you would think that they would probably be adding secondary agents or changing therapy later on as repeat positive cultures were coming off the instrument. So
Yeah, I think that's likely to be the explanation. Unless I missed it, they didn't look at an individual patient level, which is what you would have to do to sort that out. They just looked at the overall trends within each of the groups of patients.
So I read this one over briefly. Did they talk about any kind of follow-up? Did they share this data with ID, indicating, to expect longer blood culture positivity and whether that would change any downstream patient management? I don't know if they talked about any of that.
They didn't say, but the first and last authors were both in infectious diseases, along with another author, and then the microbiologists who were on the project are sandwiched in the middle for authorship on this one. So since it came from their ID group, I expect the word got out.
Yeah. Very interesting.
I do think they're important studies, as we start to implement these newer, better machines, 2.0s, to learn how to now, apply them clinically. And, there's going to be new future blood culture instruments coming down and more implementation of the virtuo systems. And so these types of studies are extremely important. And to Ellie's point, I think we need to start providing, guidance to laboratories on what to expect and, education of their clinicians when these transitions occur. And because sometimes you find things like this that you don't realize that can, although great that we're detecting more Staph aureus, it's just being interpreted in the wrong context of treatment failure or source control issues when it's just that the resin's doing what it's supposed to do in the bottle, right?
Yeah.
So
communication.
Yeah, I think that's great. I think that's
key.
the bottom line, and very well put.
Okay, so we're going to move away from bacteria for a little bit here. So the first paper I selected is by Kausic Data and colleagues, which was just recently published a few weeks ago, and it's entitled Development of an Interferon Gamma Release Assay to Aid in the Diagnosis of Histoplasmosis. And the reason I chose this is pretty straightforward outside of my personal interest for histoplasma diagnostics. I do think that any diagnostic advancements for this pathogen are really most welcome given the limitations of currently available testing. And to kind of set the stage here, the challenge with current diagnostics is that there's really no one or maybe two sizes fits all diagnostic schemes for diagnosis of histo. Sometimes to me it seems like it's more of a kitchen sink approach where we're throwing kind of a combination of multiple direct and indirect testing methods, each of which have their own sensitivity and specificity limitations that we need to remain cognizant of. And then additionally, as this paper kind of zeroes in on, while most infections with histo occur after initial exposure, the organism can sometimes hide out in kind of these latent foci and then cause disease at later time points when the patient becomes immunosuppressed. And in those latent infection or asymptomatic infection scenarios, I think current diagnostics are even less likely to detect that infection. So this group hypothesized that probing the cellular immune response against histoplasma can be an additional way to detect active and potentially latent histo infection, similar to our current approach for diagnosis of latent TB. And so towards that goal, they developed this interferon gamma release assay, or IGRA, using ELISpot technology, and they refer to it as the Histospot assay. So the way this test works is they isolated yeast cell lysate from one of the most common histoplasma strains in North America. So it's not recombinant antigen, it's kind of whole cell yeast lysate. So that's something to keep in mind. And they use that lysate as the stimulatory antigen for their T cells. And then all patient samples were also tested with a no antigen negative control and then separate stimulation with phytohemagglutinin, which is a universal T cell stimulant, and that acted as the positive control to ensure that the patient had a sufficient number of active, happy, functional T cells. So, as a refresher for how IGRAs work, essentially patient peripheral blood mononuclear cells, or PBMCs, which are primarily lymphocytes, are isolated, and then they're incubated with the antigens of interest. And if the patient's T cells have previously been exposed to the pathogen, they would be stimulated to produce interferon gamma, which in this study, that interferon gamma is detected by an ELISpot assay, which, I'm not going to go into the details, but essentially it involves counting spots, and each spot equates to a single interferon gamma producing T cell. So,
Ellie, can I
yeah,
ask you quickly,
yeah,
why would they choose an ELISpot? Like, it's more challenging than other, EIAs and other assays for interferon gamma. So what would be the
yeah.
advantage of a ELISpot?
No, great question. They are much more challenging because you do have to isolate PBMCs, and then you got to count them. And then you have, the readout again is to an extent subjective, even though there's ways to make it objective, but you count spots. However, the benefit of doing that is that you start with a known amount of PBMCs. So in this case, they determined that they need 10 to the fifth PBMCs in order for the assay to be run. Whereas with other types of assays that are based on an EIA or a chemiluminescent-based platform, there's no pre-counting of the lymphocytes. So I think it's a great idea to start with an ELISpot first to make sure that you are enhancing the sensitivity as much as possible. But they are tough assays to run, especially if it's going to be high volume testing. It's a lot of work to isolate PBMCs and keep them happy, honestly. But it's a good way to start for test development
Great,
purposes.
thanks.
So, so kind of with that background, they went into a number of kind of proof of concept studies, which again I won't go into, but they essentially established the minimum number of PBMCs they need to run the assay, incubation times, and then acceptance criteria for their internal controls. And then they also determined the minimum number of spots, if you will, that are needed for a patient sample to be considered positive, which in this case is 4 or more. And then impressively, they also did fish flow analysis, which we don't see too often in JCM. But they essentially did that to document that histoplasma stimulated T cells do specifically produce interferon gamma mRNA. So using their optimized ELISpot, they evaluated a couple of different patient cohorts, one from Johns Hopkins actually, that included 21 patients with proven or probable histo and 23 patients with possible histo, all based on EORTC MSG criteria. And then they had 34 control patients with other invasive fungal infections. And in that cohort, they found a sensitivity of their IGRA assay of 77% when the possible histo cases were excluded, but that sensitivity dropped to 64% when those possible cases were included. Importantly, though, they had 100% specificity, which was pretty cool. Again, their control patients were other individuals with other IFIs like Aspergillus or crypto. Again, all negative. But I think the one control group that was missing, in my opinion, were patients with blasto. Right. And, given the limited ability of other serologic assays, aside from maybe immunodiffusion, to differentiate between these two, I'm really curious and excited to see the next study with this assay and see if they include some blasto patients in there and see how that specificity stacks up. And then finally, the second group of patients they looked at were 44 otherwise healthy individuals from Tennessee, which is a notably a region of hyperendemicity for histo. And they found that even though these were healthy individuals, about 1/3 of them were positive on the histospot assay. And while these could be false positives, the authors suggested that these results could potentially indicate that they are latently or asymptomatically infected. So ultimately, I thought it was pretty intriguing, a promising proof of principle study. And as the authors kind of state in the discussion, this sort of assay could potentially be used to identify individuals with latent infection prior to initiation of immunosuppressive regimens, which would put them at risk or at high risk for disease later on. So yeah, I think it was a great study, potentially a nice addition to our histo diagnostic armament in the future. So we'll see where they go next. Thoughts?
That's very exciting. First thought.
Thanks.
Oh, Alex, did you want to go?
I think you're muted. Sorry, I was looking at Alex, but Trish.
Sorry, I am muted. Go ahead, Trish.
Yeah.
I was just going to say I was very excited to see this. And then, just thinking about the control group from Tennessee, where they turned up positive by the IGRA. Was there any other testing done to help differentiate between false positives, or, were they truly infected, or?
Yeah, that's a wonderful question. I was digging in the paper to find more information, and I don't know if I missed it somewhere, but I didn't see if they'd had serology done, right? For example, if they'd had previous testing done in the past, but no, there wasn't any additional kind of investigation to look at that further, but that would have been a nice addition as well to help tease that out. Is it false positive, or are they really previously latently infected?
I'm giggling inside because I feel like the answer with Ellie all the time is serology.
Hey, don't put it down.
I'm not.
But that is usually the answer, yes.
Hey, Ellie, you mentioned that we have kind of a kitchen sink approach to diagnosing histoplasma now. Would this get us away from that, or would it be one more thing we're throwing in the sink?
Yeah, I probably wouldn't get away from that. But I think there's different clinical scenarios where the different testing is most appropriate. And I don't know that I would use this necessarily as an upfront diagnostic in all cases, but I think the idea of using this to screen patients before immunosuppression, if they come from highly endemic regions, I think in those cases, it would really be a significant value add. So yeah, I don't think we'll probably ever have a 1 or 2 step diagnostic approach for histo.
Well, it keeps you busy.
Yeah, but I could be wrong. Who knows? All right, so we'll move on. Trish, what's your second paper?
Oh, I'll give you a guess. I did NGS on the first time. What's going to be my second?
AST?
I'm out. Alex, it's all you.
So yes, I'm that predictable. So my second topic is on antimicrobial susceptibility testing. And of course, it's, again, I'm excited about the whole comeback of disk diffusion testing in the lab, especially with incorporation of total laboratory automation. So the combination of both really allows us to provide more rapid phenotypic AST results, which was nicely highlighted by the paper entitled Fully Automated UCAST Rapid Antimicrobial Susceptibility Testing from Positive Blood Cultures, Diagnostic Accuracy and Implementation. This was by Charcuy and Schrenzel's group in Geneva University Hospitals in Geneva, Switzerland, which all three of us need to go for a field trip and visit them, obviously.
I'm down.
Yeah.
So we know that disk diffusion provides flexibility and a cost-effective alternative to antimicrobial susceptibility testing. And what we've seen over the last couple of years is the development of standardized rapid antimicrobial susceptibility testing, or RAST, rapid antimicrobial susceptibility testing methods, directly from positive blood cultures. And they have been developed by both CLSI here in the US and UCAST by our European colleagues. In this study, they further evaluated the UCAST RAST method from positive blood cultures, where you can evaluate your disk diffusion results within 4 to 8 hours of incubation from a positive blood culture bottle. And we know that although these are exciting alternative rapid phenotypic methods from positive blood cultures, the manual method to disk diffusion is labor intensive in the absence of automation and relies on specific time intervals to reading the results. And so it's really difficult to operationalize from positive blood cultures because your positive blood culture can go off at any time of the night, and it's something that you don't really want to be batch testing. So in this study, the group further evaluated the accuracy and robustness of fully automated RAS directly from positive blood cultures using the WASP CoPan system to define any to try to define any implementation constraints of this method. So the study was done in 2 phases. The study was done in 2 phases. The first phase was spiked blood cultures using 779 non-duplicate clinical isolates, including a variety of resistance phenotypes, including ESBLs, carbapenemase producers, multi-drug resistant Pseudomonas, Acinetobacter, VRE, MRSA, etc. And in the second component, they did a prospective study, including 534 positive blood cultures sequentially processed in routine at the bacteriology laboratory of Geneva University Hospitals. So during the prospective phase, they excluded any polymicrobial blood cultures and did not include any species for which there was no breakpoint set by UCAS for the RAST method. So using total laboratory automation, they generated several high-resolution digital images under different light and exposure conditions at defined time points of 4, 6, and 8 hours. The zones of inhibition were automatically derived by the WASP lab, but confirmed by a microbiologist or an experienced technologist. So 5% of those reads, of the automated reads, were then manually adjusted by those who were checking how the zone inhibitions were defined by the automated system. The RAST results were assessed against automated UCAS standardized distribution testing from recovered isolates from the positive blood culture. I thought it was interesting in this study that the authors also applied a set of discs covering all the species analyzed in the study to reduce the turnaround time. So they would automatically set up their discs from the positive blood culture, no matter what it was, because they had a standardized set of discs, without needing to wait for a gram stain, a MAL-DID, or a molecular result to kind of tailor what discs were being applied. So overall, they found that the readability of the zones were improved with total lab automation of this approach, and that for E. coli, Acinetobacter, and Klebsiella pneumoniae, the great majority of zones could be read at 4 hours using the automated approach, and increased to 100% at 6-hour reads. For Pseudomonas, the proportion of readable zones at 6 hours was 97% and attained 100% by 8 hours. And not surprisingly, the gram-positive bacteria required more time to provide readable inhibition zones, but achieved 100% of readable zones at 6 hours. For UCAS methods, they introduced this interesting category back in 2017 called the area of technical uncertainty. And this concept is an area of technical uncertainty for bacteria and antimicrobial agent pairs for which the interpretation of routine susceptibility results is uncertain. So it's kind of this gray area with AST. It's not a perfect method, and UCAS acknowledges that through this ATU. And so they found that with the shorter incubation periods from the disc diffusion, you've seen a higher number of results being interpreted as falling within the area of technical uncertainty. However, if you let it incubate a little bit longer to the 6 or 8 hour, extending that incubation period, those zones were no longer falling within that ATU and allowing for interpretation of those results. But overall, the categorical agreements for all species analyzed were greater than 95% at the different time points. And the results from the spiked blood cultures were predictive of the prospective blood culture testing. They were really, the results were hand in hand and followed the trends that we've seen with the spiked as they were in the prospective study. Although the performance was great overall, there were some differences. So Klebsiella pneumoniae, all zones could be read at 4 hours with greater than 98% categorical agreement across the agents tested for the various time points. Whereas with Pseudomonas, which is usually the problem child with rapid phenotypic methods, raised several challenges. The first was that there was lower categorical agreement for imipenem at 6 hours, and it didn't improve with prolonged incubation. And so the authors actually do not recommend testing imipenem against Pseudomonas aeruginosa by this method due to those results that they encountered. They also found specifically with Pseudomonas aeruginosa that piperacillin tazobactam, ceftazidime, and cefepime cannot be released at 6 hours and needed to be incubated to the 8-hour period to be released reliably. So, but overall, the results of the study established the performance of a fully automated UCAST rapid AST method directly from positive blood culture bottles to be consistent and robust with our standard disk diffusion methods, and can even detect many of our more resistant organisms. And the automation, using this automation method can markedly enhance the percentage of readable inhibition zones and reduce the percentage of isolates categorized in that area of technical uncertainty. And so a fully automated UCAS RAS method can substantially improve laboratory workflow by reducing the hands-on time and removing that strong constraints linked to manual readouts at precisely defined times, because you can just program your system to take the pictures at the right times, for example, or actually set additional rules, like with Pseudomonas, that you can't read the ceftazidime, cefepime, and piptazo at 6 hours, and it needs to go all the way to 8 hours, where, if you did this manually, your technologist would have to remember all the exceptions and all the rules. And so it's kind of nice of further utilizing total laboratory automation in our hands, and also do it in a more cost-effective way with the comeback of disk diffusion. I do want to highlight one last study before I pass it on. And this is a study that was entitled "Stop Waiting for Tomorrow, Disk Diffusion Performed on Early Growth is an Accurate Method for Antimicrobial Susceptibility Testing with Reduced Turnaround Time." This was from Dr. Weber and Dr. Carrian's group at WashU. And they actually found that, the common recommendation is not to set up disk diffusion unless you have a 18 or 24-hour subculture. And they looked at just taking earlier subcultures at 6 or 10 hours to see if you could still reliably reproduce your disk diffusion results. And they found that, yes, you can incubate for a short 6 hours your isolate or subculture prior to setting up your disk diffusion, and you don't need to wait till tomorrow. The one thing I love about this paper is that it's a simple question, and it really challenges some of the dogmas we have. It doesn't need to be expensive molecular techniques that we publish on. And the second thing is the inspiring titles. My titles are so boring on my manuscripts. I'm like, why can't I think of fun things like this? I feel like their group is always very good at coming up with clever titles.
Well, something to aspire to, Trish.
My goal for 2023.
Yes. So a quick question for you. So you and Alex and some of our other listeners out there know that I don't necessarily follow the AST world as closely as some. So bear with me. But can you quickly talk about whether there's any differences between CLSI and UCAST rapid AST methods?
Very quickly, yes, there are differences.
Okay, good. Thank you.
That's the first thing to know, and that you can't mix up using the interpretation of one over the other. That if you're using the UCAS method, you need to use the UCAS interpretive tables versus if you're using CLSI, you need to know CLSI. There's differences in how the inoculum is set up, the organisms, the drug-bug combos, the reading times, etc. So very shortly, that's my answer.
That's a great answer. Perfect.
I think those are both really interesting studies. And on the second one, the one you mentioned briefly, to me, Carrie-Ann Burnham publishes on lots of things, but to me, that's a classic Carrie-Ann Burnham study. She saw a question that was right out there that nobody else noticed, right? She saw dogma and she questioned it. And then, as you pointed out, she had a great title for the article as well.
I completely agree, Alex. It's a great example for all our fellows too, right?
Yes.
Like we don't need to think outside of the box. There's questions that we're looking at every day that we can publish on and try to answer different questions.
Awesome. Well, Alex, you want to share with us your second choice?
All right. My second choice was a paper titled Detection of Newly Secreted Antibodies Predicts Non-Recurrence in Primary Clostridioides difficile Infection. First author is Natalie Haddad. Corresponding author and last author was John Dace. And this is primarily out of a company called MicroBplex, and they worked with a number of collaborators at some of the Emory hospitals. I chose this for a couple of reasons. First, it includes novel technology, which I always find appealing. And second, I have a very soft spot for immunological tests of all kinds because my PhD was primarily in immunology. I moved into microbiology later.
It's the
So
best area, Alex.
at the risk of going into Ellie's lane, I'm going to try to talk about an immunology paper. You can stop me when I get it wrong. I think everyone knows C. difficile infection is common. The burden in the United States is nearly 500,000 cases per year. Recurrence of CDI, Clostridium difficile infection, is also common, occurring in about a quarter of people after their treatment for their initial bout of CDI. The authors suggest that predicting the risk of recurrence could impact therapeutic decisions, such as the use of fecal microbiota transplant. And the earlier you could predict recurrence, the better, because then you could adjust therapy before the recurrence happens. So let's start with the underlying technology. They developed an assay to detect antibodies that were being produced by antibody-secreting cells in the blood. Most antibody-secreting cells in the blood are so-called plasmablasts. These are cells that secrete antibodies, and then most of them die by apoptosis, although a small number go on to be long-lived plasma cells. The idea is that we might be able to detect antibody responses earlier by detecting them from antibody-secreting cells rather than doing testing on serum. And on the company's website, they also suggest that antibody-secreting cells could indicate current infection rather than past infection, but that's not explored in the paper I'll discuss. So the assay is called MENSA, M-E-N-S-A. And what do you think of when you think of MENSA?
I'd rather not say. Is there something else?
How about you, Trish?
I can't follow up with that.
Oh, so I usually think of the group of people who are allied by their high IQ, but it turns out it's from the Latin.
Not what I thought.
It's from the Latin for table. I don't know what I do know what you two are thinking, but it's from the Latin for table, and so I don't know why they're named their assay after that. But MENSA in this case stands for medium enriched for newly synthesized antibodies. And basically what they do is purify lymphocytes from peripheral blood, wash them repeatedly to remove antibodies from the original sample, and then put them into cell culture with conventional media for 24 hours. No more giggling. There's no antigenic stimulation provided during the culture. Then they collect the supernatant and they assay it for antibodies that are produced during that 24-hour culture. This was a small study. I would characterize it as proof of concept, not a full-blown clinical validation. The control group was 64 people. The CDI group included 46 patients, 37 of whom did not have recurrence and 9 who did, as defined by diarrhea within 60 days after treatment and a positive C. difficile assay. The blood samples were collected from the CDI group at various time points and were used for MENSA testing and for testing of whole sera. MENSA or patient sera were tested for antibodies to a panel of 10 different C. difficile antigens. These included different domains of the C. difficile exotoxins, as well as other antigens such as flagellin, cell wall protein, and glutamate dehydrogenase, all from C. diff. Antibodies were detected using the Luminex platform, which is a well-established system for multiplex serological assays. And as has been seen in other studies, patients with recurrent Clostridium difficile infection were more likely to have antibodies against patients with recurrent infection were less likely to have antibodies against C. difficile antigens. Antibodies for most of the antigens were undetectable or very low in those patients with recurrent disease. In contrast, 70% of the patients who did not have recurrent Clostridium difficile infection had antibodies detected in either the Mensa assay or by serology, conventional serology, often with high fluorescence values in that Luminex assay. When they looked at the different antigens, they found that 3 of them were really most useful in distinguishing patients with recurrence from those without recurrence. They were a domain of toxin B, a domain of the specific form of toxin B produced by the hypervirulent C. difficile, and a domain of binary toxin, again produced by hypervirulent C. difficile. And hypervirulent C. difficile, just to remind everybody, is a specific lineage of C. difficile named NAP1/B1/027, and obviously it's associated with more severe disease. Positive results with at least one of those antigens identified 88% of Mensa-positive non-recurrent patients and a similar proportion of serum-positive non-recurrent patients. So the big question is whether Mensa performed any better than conventional serology. And I would say the preliminary answer is yes. For the 3 antigens I just mentioned, there was a 19-fold greater relative risk of recurrence in Mensa-negative patients than in Mensa-positive patients. Using serum results, the relative risk was 13.6, so Mensa was a slightly better predictor. Both of these assays were much better predictors of recurrence than clinical characteristics like age, white cell count, and antibiotic use at various times and for various reasons. They also looked at when the antibodies were detectable in patients who did not have recurrent disease, and they found that a larger proportion of patients had antibodies detected early by Mensa than by testing of serum. So for example, 2 to 6 days after initial onset of symptoms, 55% of non-recurrent patients had antibodies detectable in Mensa, and only 41% had them detectable in serum. At 7 to 12 days, it was 63% positive in Mensa and 54% positive in serum. At later times, the 2 performed equally, where serum was better. Those differences are not large, and it would be helpful to see if they hold up in a larger study. And I'm going to just summarize the last finding here, which was that it turns out that the Mensa assay is very effective at primarily detecting IgA and IgG, as opposed to serology, which was primarily detecting IgG. And they found that IgA was the best predictor of whether or not the patient was vulnerable to recurrence. And that fits because we know that IgA is the antibody class that's secreted into various body sites, including the gastrointestinal lumen, where it could neutralize the toxins produced by C. difficile and presumably prevent their effect on the intestinal epithelium. So I think this is an interesting preliminary study. Clearly, a larger study is needed to more robustly assess the ability of the assay to predict who will or who will not get CDI. So Ellie, how did I do with immunology?
A+. Well done. Yeah, I actually, out of the 2 studies that you chose, this is my favorite one. But I thought it was really cool, but man, isolating PBMCs, like nobody really wants to do that on a routine basis. And
Agreed.
I know I'm going to be getting into the nitty-gritty here, so like, sorry. But I was looking at their methods, and their serum, they ran it at a dilution of 1 to 1,000, which may not mean a lot for some of us, but typically when we do serology, we run it tenfold lower at 1 to 100 dilution. So I'm kind of wondering, if they brought down their serum dilution tenfold, would their sensitivity early on increase, which would be nice. But again, not to take anything away from their study. Really, really cool. I did have one other thought, so more really a comment than anything else. Pfizer has a C. diff vaccine, or had, and it was targeting modified versions of toxin A and B, I believe. And they got to phase 2 or phase 3 clinical trials, but they ultimately had to pull it because there was no benefit to protect against primary C. diff infection. But yet here, they show that the presence of antibodies decreases the risk of recurrence. So that's kind of interesting. And maybe there's still hope for a C. diff vaccine in the future. So that was more of a comment than a question, but I thought it was a really cool study. So
Yeah, I was very excited when I read the title of that one. I haven't hadn't picked that one up yet. And so, but I agree, it's prelim data, right, that we need
Yep.
further studies
Yep.
to evaluate it. And I think they need Ellie on their study to help with their dilutions.
Well, yeah, just that different dilution factors is all. All right, so I will switch gears and we will go to the last study we're going to talk about today. So this paper is by Ndama and colleagues and is entitled Accuracy of Tongue Swab Testing Using Expert MTB-RIF Ultra for Tuberculosis Diagnosis. And as for why I chose it, I'll be perfectly honest. It was simply the sample type that caught my eye initially. And, we don't really specifically call out tongue swabs for other clin micro testing. And then I saw a second paper on tongue swabs for TB published in JCM, and I thought, hold up, I got to look more into this. And so here we are. So I'm sure some of you are thinking, why tongue swabs when we have sputum, which is a much more optimal sample type, really well-established sample type for diagnosis of pulmonary TB. And this is where I really commend the authors. I thought they nicely focused on the fact that some individuals, like kids and people living with HIV, frequently do not produce sufficient sputum, even though, as a dear friend pointed out, well, you can induce it. Yeah, but that's no fun, right? And
this is a big deal in pediatrics.
Yeah.
A big deal.
Yeah. Right. And then sputum collection can also be hazardous for healthcare workers in areas where safety measures are maybe not as readily available or adequate. And then sputum is just not an ideal sample type when the goal is to actively screen high-risk populations, as is not infrequently done in high TB burden areas of the world. So overall, this sort of approach of using simpler tongue swabbing would likely have a larger role to play in more under-resourced but higher risk patient populations. So with that background, this group really set out to optimize tongue swab processing for use on the Expert Ultra MTB-RIF assay, because previous attempts, and yes, there were previous attempts to do this, showed sensitivities in the 40 to 50% range. So this group used special Copan flock swab tongue swabs, which, quote, have been shown to increase tongue dorsum biomass collection. And they evaluated 3 different collection and processing methods. So using either a single tongue swab or 2 tongue swabs placed in Cepheid sample reagent and then extracted the same way that you'd extract sputum samples. And then their third method, they used a single swab, but they didn't put it in sample reagent. Instead, they boiled it in a different buffer. So using these 3 methods, they started out by swabbing TB negative individuals and doing just some MTB spiking LOD studies. And they ultimately found that the boiling method had the lowest LOD, followed by the double swab method. But they ended up proceeding with the double swab method because it didn't require boiling and it more closely followed the sputum prep process for the expert assay, which would ultimately be easier to implement. So to evaluate this double swabbing tongue method in a real-world population, they enrolled 183 adults presenting with at least 2 weeks of cough to various healthcare centers in Uganda. And they collected 3 tongue swabs, with 2 of them placed in a single tube, and then 3 sputum samples collected after tongue swabbing. And then the sputum was tested by the MTB-RIF assay, but if it was negative by that test, it was cultured in liquid media, or if that was not available, cultured on solid media. So amongst their patients, 54 were positive by the MTB-RIF assay on sputum, with the dual tongue swab samples positive in 78% of those patients, with 100% specificity in the TB negative group. An additional 4 patients were culture positive, though, which brought the ultimate sensitivity down to 72% when culture and the Expert Ultra were used as the reference method. And then, not surprisingly, when looking at the semi-quantitative sputum results by the Expert Ultra, the tongue swab method missed patients that resulted as trace or very low quantities of TB, which obviously remains problematic for HIV patients and for kids, again, who have lower TB loads than other patient groups. So overall, I thought the group showed significant improvement in the sensitivity of tongue swabbing over prior studies, which, I mean, I thought it was pretty cool and encouraging. So we'll see where this goes. I mean, I don't know, maybe we'll all be swabbing tongues for TB 4 to 5 years from now, right? So yeah, I thought it's a really interesting approach, I guess. So
I love how they're trying to develop it further too. Like there's, if there was already publications on it, they're like, what can we do to, increase the sensitivity? Because that's obviously the issue here.
yep.
Is there anything else they can try, Ellie, like at this point?
Yeah, so they are looking, one of their main goals is to look at alternative swabs that collect more tongue biomass, which, kind of sounds, I don't know how they're going to do that, right? Just like keep scraping the tongue. So that's one approach. And then the second study I mentioned that had, that looked at tongue swabs actually paired tongue swab testing by molecular means with the lipoarabinomannan, lamb. We'll go with lamb antigen in urine. And found that, combining those two non-invasive testing methods still didn't reach as good as like sputum testing, but it did offer some improved sensitivity. So I think there's a lot of interest in continuing the hunt, continuing the search for non-invasive sample collection types for TB diagnosis. So we'll see where they go.
I really applaud the authors. I think this kind of work in making sample collection easier is really important. It's easy for us in the lab not to appreciate that patients will skip appointments, patients will, do what they can to avoid getting stuck by a needle, to avoid invasive things like bronchoalveolar lavages. So I think this is terrific. It increases accessibility to testing.
Absolutely.
Great.
All right. Well, for our listeners, you might have to listen to this episode in 2 parts. But we hope you enjoyed our selected papers. And Trish, thank you again for joining us. It was a pleasure. We learned some stuff. We laughed.
Thanks for having me again, and happy holidays to all the listeners. Special shout out to a few of you. You know who you are.
And Alex, any closing words or thoughts of wisdom or words of wisdom?
No, it's been a great year for the podcast, and I'm looking forward to the next one.
Same. Well, I hope everyone listening has a happy and healthy holiday season, and we will see you again in January. Thanks, everyone.